Thanks, Matteo, to gave a really nice talk before. And today I'm going to be the bad guy. I mean, because as a scientist, we need to be aware about limitation of these techniques that we use every day, and if it's possible to overcome them. The idea of this talk is to give you a brief overview of the limitation of tractography specifically, and show you some methods that we can try to use it to find the right pathway in this jungle of streamlines. So, okay. So I'm going to talk about why we need to validate tractography approach. I will show you, as I said, some methods to validate tractography and fiber orientation estimation, such as software and hardware phantom, postmortem and biological samples, and challenges. With challenges, I mean the recent efforts that the scientific community made to test and validate tractography and diffusion-weighted imaging. And of course, at the end, I will summarize the main findings that we learned from that. So we know that diffusion imaging is probably one of the, oh, this should be a video, but okay, perfect, it's not working. The diffusion imaging is probably the only method to study microstructure in the human white matter. And with that imaging techniques, we can also calculate main trajectory and major axonal pathway. The first problem is it's for sure that we don't have any gold standard rules. Each group uses their configuration to perform tractography. They calculate DVI indexes and so on. Differently, with the animals, we have some gold standards configuration, like approach. Like we can use neural tracer and see exactly the axonal pathway through the white matter. The invalidity of tractography in humans came from different sources, such, for example, partial volume effect or crossing fiber issues. We also have to deal with other noise sources, such, for example, MRI artifacts and motion artifacts. And we have also to decide, when we approach diffusion imaging, we have also to decide, for example, which is the best way to model fiber orientation. There are plenty of algorithms that we can use right now. And also, as Matteo mentioned, we have a lot of criteria that we can use, especially in deterministic tractography, like, for example, define a certain amount of value of fractional anisotropy to stop the streamlines. So, why we need to validate? Well, it's science, of course. Scientists are continuous to modify, correct, and rebuild new theory that should be better explain the real biology, the real ground truth. I don't want to say anything bad on fMRI, for sure. But even this methodology that has been used for decades studying the human brain suffer from major controversies, such, for example, we still don't understand why it's necessary for our brain to increase the flow response after we deliver, for example, in the fMRI, we deliver a certain stimulus. One of the first methods that has been used to validate and to validate tractography and fiber orientation estimation is the software phantoms. Basically, the software phantom is a mathematical model that simulates diffusion signals with a well-known ground truth. With this simulation, we can test if the fiber's orientation algorithm is good to estimate the complex fiber geometry, and if tractography algorithm can track the right fiber bundles. However, there are some limitation on this approach, even if it's extremely flexible. Sometimes it's often to find some models that are basically constructed to show that a given algorithm is really good, and this software phantom also is so far away from the real biology of the brain. We don't know exactly how we cannot reproduce and mimic how a certain brain configuration can influence the diffusion weighting signal, for example. Another method that has been used in this field is the hardware or physical phantoms that provide basically a different balance between a ground truth control and the real signals. In fact, it's possible to control the factor that affect anisotropic water diffusion using, for example, different materials, and using also different fiber configuration. You can see region that we can use to estimate fiber orientation. However, we are still far away from real biology, and we don't know right now how to perfectly mimic axonal properties in this kind of phantom. To move close to the real biology of the brain, scientists started to look at the postmortem tissues. It's not possible to control everything in this kind of sample, but this biological phantom provides physical properties that are close to the neural tissues. With this approach, it can be possible, for example, to scan the sample for a very long time, like days, weeks, months, without making anything bad to the sample. And also the other advantage is that we don't have any motion artifacts and other artifacts related to the MRI acquisition. Another important advantage that we can use, we can obtain histological information from the tissues. And so we can compare this information with our tractography. Here I want to show you a classical example of post-mortem dissection. In this case, the goal was to identify a real tract in the post-mortem brain, the human brain, and compare it with the results of the diffusion weighted imaging on the healthy subject. So this procedure involves a series of steps in which the brain needs to be fixated, then cleaned from non-brain tissues, some like arachnoid membrane and vessels. Then the refrigeration step, it's really important because allow to better identify axonal pathway causing axon to swell, and help also to remove gray matter cortex. Then, after we've done these steps, we can start to dissect the brain and identify major tract and use its tools, we can extract from the brain the tracts. Like here, we can compare this tract with the tractography results and see how they different. We can also compute and calculate false positive and overlap ratio and other many index that tell us how confident was our algorithms respect to the real biology. Another example of post-mortem the biological phantom is from Catani's group, that they study geometry accuracy of DTI tractography and DTI indexes in ex vivo human cerebellum with a super resolution of 100 micrometers. With this sufficient resolution and signal, it's possible to say that, and it's possible to see that DVI signals, like here, okay, thanks. DVI signals and DVI indexes, sorry, can differentiate from the gray matter, the third layer of the gray matter, and the first one, and the white matter. And also tractography can reveal some precise pathway related to granular cell and T junction in the cerebellum. While this technique is promising, we have to consider some limitation. For example, fixating the brain modify water diffusion, which inevitable change some biological properties of the tissues. The tissue relaxation is shorter, and we need custom sequences and coils to able to achieve some signal. Also, the tissue preparation require an incredible manual skill, because there is a high risk to modify this inner geometry. For example, by squeezing the brain or just moving the brain to one place to the other. The time needed for scanning is incredibly long, and I'm sure that your colleagues will sure complain about it, because you will occupy the scan for days and weeks. And the operator has to have a precise knowledge of the neuroanatomy, and this procedure is still subjective on human error during the dissection procedure. So, due to the relevance of fiber tractography to investigate human function, one way for sure is to validate, one alternative way to validate tractography is to ask the scientific community to participate at a public organized challenge. In this way, for example, an algorithm or solution may be developed to address a specific challenge or specific issue in this field. Now, in this section, I'm going to show you the major competition in this field and their goals. I'm not going to show you final results of this competition, because I don't have time to explain in details each competition and their results. And also, it's your duty as a scientist to find optimal way of to address your research question. So the first one, FiberCop, was the first community effort to try and get some quantitative evaluation of tractography algorithms on a common dataset, on a physical phantom. This challenge was to find 16 existing connections on this phantom, and using a predefined ROI, a voxels ROI. So you can see here the ground truth is well defined with some fanning fibers, kissing fibers, configuration, crossing, and curvature, and so on. So the participant has been, they deliver this phantom to the participant and ask them to calculate and use their algorithm to perform tractography. In this example, you can see here, like, how can I, no? You can see here, I don't know if you can see, but there are the 16 voxels, ROI voxels. Below, it's the ground truth that we expected to find, basically. They expected to find the fiber. And here, you can see some results. For example, in the upper part, on the left, in this position, you can see that all the algorithms track the right fiber bundles. However, one was incorrect. There are some limitations in this challenge that we need to take care. First, this challenge was limited to deterministic tractography, and also, as I mentioned, these physical phantoms differ from living tissues, and diffusion here occurred only in the extracellular compartment. One really nice aspect of this competition was that the researcher didn't know the real fiber geometry of these phantoms, so they basically tracked the streamlines and hoped that was in the right position. Another interesting challenge was the DTI challenge. That objective was to reconstruct the best corticospinal tract as judged by a panel of experts. The corticospinal tract was chosen because it's one of the well-known tract in human that has its anatomical origin and termination. Interestingly, the data, these are real data. The data also came from patients with glioma that cause, near the corticospinal tract, that cause partial volume effect and distortion and volume distortion in one of the cortical spinal tract. So the judge can also distinguish and see by compare the healthy corticospinal tract with the not healthy corticospinal tract. They compare and validate tractography. So the judges have to decide about accuracy of the submission and also about the speed and reliability of the process. They basically ask the participant to pre-process and estimate tractography in front of them at the conference. I don't remember which one, maybe Mikai or don't remember. Another real interesting challenge was the tractography challenge. The goal was to provide the best possible reconstruction or set of fiber pathways in a realistic simulated replication of the whole human brain diffusion that are with properties that are typically of a clinical acquisition. So this was achieved by manually estimating and segmenting 25 fiber bundles in a healthy subject. They use one subject from Human Connectome project. Then this estimation was used to create a ground truth background for creating a synthetic fiber bundles. You can see here all the steps that they have done. So segment a certain fiber bundles, then use it as a ground truth for creating a synthetic bundle model, and then combine these 25 tract to create the wall diffusion brain. And they ask for sure to the participant to estimate these 25 fiber bundles. The results are quite impressive. I just show you this slide that it's that show only a few results of the huge research. So basically you can see they evaluate valid connection versus invalid connection, and also some measure from volume-based of bundle overlap and bundle overreach. Here, this percentage indicates the overlay ratio, and this one the overreach ratio. You can see, for example, on the cortical spinal tract that DTI-based deterministic tractography covered overlay 20% of the cortical spinal tract, but the overreach was the 3%. If you move to a probabilistic approach based on the RD acquisition, you can see that overlay is increased, while also the overreach. So overreach means that the fiber bundles that are outside the ground truth bundles. Another challenge was the RD reconstruction challenge. This challenge focused on validating the local fiber orientation distribution, finding which algorithm was the best one to local estimate single and fiber and crossing configuration. The results varies from the algorithm that they use to calculate this fiber orientation, that range from the standard DTI. You can see here the ground truth of the fiber in one voxel, and here you can see the fiber estimation model from DTI or Q-ball imaging, constraint spherical deconvolution, and so on. So this challenge was just to try to find the best, the optimal algorithm to reconstruct the fiber orientation. Another challenge was the tractography reproducibility challenge with empirical data. The goal was, of this challenge was to assess the reproducibility of emerging pipelines that has been used in the clinical practice. So they asked the participant to track 10 white matter bundles on a single subject data that came from 2 different scanner, and in each scanner they perform 5 runs of diffusion weighted imaging. Each tracks here was evaluated using a dice coefficient to look the coherence of tracks within session, across session, and across scanner. The last but not least was the really recent the 3D validation of tractography with experimental MRI. This challenge was based on 3 different validation strategies. One using a physical phantom with 16 fiber bundles, an high resolution ex vivo macaque dataset with an atlas of non-connection, and also an ex vivo squirrel monkeys with the histological tracer injection. The challenge was to estimate the ground truth bundles and connection in all database. Here is just an example of one results with different submission. You can see here it's quite different from that one, and so on. They evaluate pipeline on the to process diffusion weighted imaging, algorithms, and fiber orientation. So what we learned from this challenge was that this is a really good news, that diffusion tractography is able to reconstruct valid fiber bundles, and these connections are typically very close to spatially to the ground truth. However, there are some limitations that we need to take in account. For example, when our algorithm finds crossing fiber region, that sometimes there are some incorrect streamlines that we can find and track incorrect streamlines. We unfortunately have a low overall bundle coverage and low % of valid connection. Another limitation is that use tractography in neurosurgical decision is very challenging, because we have a large variabilities among algorithms, and the present we have a huge presence of false negative pathways. Another good news that we are really good to estimate local orientation. So our algorithms right now are able to detect different fiber configuration, and the tractography is reproducible. Sorry, but algorithms can give you a very different results. Thank you. I don't know how to. Wait a sec. Sorry, just need to
Try plugging.
Yeah, I don't know why it's so fucking small over there. Look.
Oh, we don't have a... No, it was me.
Vicente, ¿puedes venir? ¿No tiene conexión a internet? Para
Just one thing, if you have downloaded the presentation in PowerPoint, please delete it. I will upload a PDF, okay? The one you see.
Dime. Dale ahí. Eso es.
Okay.
Y cuando salga lo del brain, le das a un botón que pone hacer.
¿No hay acceso al repositorio aquí?
¿Eh?
¿No hay acceso al repositorio directamente aquí?
No, tienes que hacer. Pero no, yo no lo tengo en el repositorio. ¿Lo tienes tú? ¿La tuya? ¿La tuya me la pasaste? La presentation.
Sí. Bueno, te lo he pasado hace 3 o 4 horas cuando te lo dije.
¿Qué?
Te lo he pasado hace 3 o 4 horas cuando te lo dije.
No sé si la tengo.
No,
Si no, bájatela tú.
pero es que no sale nada. Que no sale lo de la conexión.
Ay,
¿Dónde se pone eso?
un segundo. Esa de ahí. No, pero creo que ya tienes internet, ¿no? Si tienes internet. Deberías poder... Pues sí.
Con el
No.
firmware.
You have it connected to the BCBL network or eDROM, do so also. Because we are too many people right now.
Internet open.
A ver... Joder.
Que no le carga de internet.
The internet doesn't work and I don't know why. I'm gonna call Duguti.
Which internet?
The one from the Brainhack
Network, but the thing
I
is
see.
that he needs that one.
Let's go for it.
Don't worry. Yeah. So are you planning to go somewhere? Okay. Sorry guys.
No va, ¿no? Guti, se me ha desconectado el Brainhack-Donostia2019 del ordenador principal y no va el o sea, no le deja loguear. ¿Sabes? O sea, yo me conecto desde el ordenador que me dejaste al Brainhack 2019 y cuando va a intentar loguear no carga la página web. Sé que hay un problema. El ordenador o bueno, puedo intentarlo. He reiniciado la conexión. Vale, reinicia el ordenador.
Ok.
Vale, pues vamos a probar a ver y si no... Vale. Joder, hecho, estas cosas surgen cuando menos te lo esperas.
Si no pongo mi ordenador, ¿eh?
Sí, también. Total, ahora no tenemos el streaming de la pantalla, así que podemos hacerlo desde el tuyo. ¿Tienes instalada Virtual Machine?
Bueno, no, pero no la necesito realmente. Lo que pasa es que va a ser un coñazo ahora ponerlo.
Si quieres, haz la presentación con tu ordenador mientras tanto.
Espérate, si va, lo hacemos aquí. Si no,
Vale.
cojo mi ordenador directamente y lo hago todo ahí, que va a ser mejor para el trap y para todo.
Total, si es que... Venga. Ahí estamos. A ver. Wifi. Joder, cállate, coño.
Que vaya mierda el ordenador andado.
El que había. Pero sí, contiene, a ver. Esta es la primera... Mira, dale, ya está. A tomar por culo, ya lo tienes.
A ver. CDL. ¿Cómo se pone la arroba aquí? La arroba.
Arroba, tío, shift arroba. No hace falta que... Con el username basta, tío. No hace falta que pongas el arroba. Mira, tú puedes dar cuenta que... ¿Nunca lo has visto así? Pues...
Ok.
Vale. ¿Puedes mandarme el correo ese directo a mí?
Te lo he mandado a ti ya.
Sí, pues no sé si lo tengo. No lo tengo. O sea, ¿me la acabas de mandar ahora? ¿Seguro?
¿Eh?
¿Me la acabas de mandar
Te lo he
ahora?
mandado hace un rato. No, ¿por qué no se ve hoy?
Porque... Espera. Ponle...